wonder how primers and other oligonucleotides actually get made? It's this process called solid-state synthesis using these phosphoramidite monomers. So let me explain how this works. It's actually really cool, it looks kind of scary, but all you have to do is remember that basically you want to protect the parts that you don't want to be reacting, expose the parts that you do want to be reacting, and do this over and over and over again until you get the whole sequence that you want. And then you gotta clean things up and things. So let's look at a little more detail. Say you want to write the sequence CGA, how your cells would do it, and how it would happen if in like a PCR reaction, is that it would be helped out with an enzyme called DNA polymerase, and that DNA polymerase would be piecing together nucleoside triphosphates, so these individual DNA letters based on a template, so a complementary strand of DNA telling it what to add where, and it's going to be adding them from the 5' end, we say from the 5' end to the 3' end. So where the 5' refers to having this free 5' triphosphate group, and the 3' end referring to having this free 3' OH. And those two groups are then going to be what join together with the release of these phosphate groups to provide energy. And so this is going to be how your cells would write CGA, or how that DNA polymerase in the test tube would do it. And again, that's based on a template, and it's going 5' to 3'. What happens is if you get this chemically synthesized, is it's actually written in the opposite direction. It's written CGA. And instead of using normal nucleotides, so these normal DNA or RNA letters, DNA in this case, it's actually going to do it in the opposite direction, using these phosphoramidite monomers. So it's basically going to start with an A, then add a G, and then add a C. But these A, G, and C are all chemically modified. And they're chemically modified in ways that are going to make it so that we can control what reacts where and when. And so basically we can spoon feed them one letter at a time. So one of these monomers at a time, but we have to do so very, very carefully so that they don't react in ways that we don't want. You see, the reason why those DNA molecules are able to link up isn't just because there's that enzyme there. An enzyme can only kind of lead a horse to water. It can't make it drink. So it can't make a reaction happen that wouldn't normally happen. But it is able to bring together everything in the right orientation. And it's able to bring the right letter to be added in the right orientation and hold things all in place with the help of metals and things like this to make this reaction exactly happen exactly where you want, so it's not happening someplace else. But so if those molecules were just freely floating around, well, then you're not going to have these random reactions happening in your cells because the concentrations of these aren't great enough and they're not in the right orientation to react in just the right way unless they have the help of that enzyme. So if we want to get these DNA letters to join together without the help of that enzyme, well, now we're going to need to bring them together. We're going to have to like flood them with high concentrations because you don't have them all being brought together by this enzyme. And we're going to have to use these kind of extreme conditions, have things like acids and things that would not be good to happen in your cells or even in your PCR test tube. So because we need to make these extreme conditions, we need to make sure that they're not going to cause other things to react as well. And so basically what we're going to do is we're going to protect all of the parts that we need to hide, so that the parts that we don't want to react, we're going to protect them with various chemical groups, and we'll get more into what some of these are. Don't be scared off by the big names. Basically, we just have ways in which we can use chemical terminology to describe the structures of these various groups that we're using. But for now, just think about them as kind of being like gloves and boots that are shielding the parts of the DNA or RNA base, or I'm using the term base because we often use that, but really the base is just this part, and the whole thing would be your nucleotide. But in this case, instead of a nucleotide, we have this phosphoramidite base, where this phosphoramidite is basically referring to this thing you have, this phosphate group, and then it's an amidite. So it's an amine group of it. And so we'll get much more into all of this various stuff. But The point is that we're using these protective groups in order to shield the parts of the molecule that could react when we don't want them to react. And then we deprotect them, so we expose them, we take off that boot or we take off that glove when we want them to react. And so in this way, we're able to carefully choreograph the addition of those individual monomers, so those different letters in the sequence that we want, in the order that we want. And there are also steps that we'll use in order to get rid of things that we don't want. And so you kind of go through this cycling of having these protected, having all these groups protected, and then deprotecting the ones that you want to react at, changing the conditions so that the reaction is going to happen, and then changing the conditions so that that reaction won't happen anymore, and that anything that is like wrong will be chemically modified so that you don't accidentally add on to them. So let's think a minute about the types of things that we need to avoid. So with that just simple example of CGA, you can see that there are a lot of things that can go wrong. So basically, if you start with an A and then you add some G's, you can get what you want, where you have this AG, or in the 5' to 3' direction, we would say GA, or we could have it uncoupled, and so where we don't get that G added. And this is something that we're going to need to remove, or at least stop from reacting again, because if you were to then take this uncoupled product and you were to go and you were to add some C, well, now what could happen is that if it was unprotected, this A could then bond to the C, and this is going to give you this AC sequence, which is not what you want. Additionally, if you didn't have this group protected during the step where you were adding G's, you could have multiple G's be added, and you can also have multiple C's be added and things like this. So there's a lot of different things, different sequences that you could end up with if you didn't have those reactive groups protected. And so we're going to need to protect those reactive groups. Additionally, in the case of the RNA, we're going to have to protect that 2' OH group, because what can happen is this can react with things, or it can actually kind of kick off the other parts of the chain. So the reason why RNA is less stable than DNA inherently is that it has this 2' OH, which is able to do things like attack this phosphodiester bond, causing chain breakage or chain shifting. And so we need to protect this 2' OH group in the case of the RNA. And so there are different chemical groups that we can use to protect these various parts. And these groups are chosen in part because of how we are able to selectively add and remove them. So these are some of the most common groups that are used are like DMT in order to protect this 5' position, and as well as we use like TBS often to protect that 2' OH. And then that 3' position, that OH, is often protected by 2-cyanoethyl diisopropylamino. So don't be too scared off by that name. Remember, those are just characterizing the different chemical groups. It's a long word, but really you can just think of it as kind of like a shoe that's on this reactive group. So it's going to prevent this from reacting when you don't want it to react. One thing you might realize is that we don't see any phosphate groups at this 5' position. Instead, the only place where we see a phosphorus is actually in this 3' position. Because when those bases are getting added, when these letters are getting added, these phosphoramidite monomers, they're actually added not with any phosphate groups on them, but with a free 3' free 5' OH instead of a free 3' OH, which is weird because normally in your nucleotides, you would never have a 5' OH. But in the case of these phosphoramidites, we do have that OH at that 3' position. And this is where you're going to get addition to. And it's going to attack here so that you end up with this phosphodiester bond after an intermediate step. So we have the 5' without any phosphate groups, but with a different group. And so this different group is going to be this DMT, this 4,4-dimethyloxytrityl. And so we'll see later, we're going to have this detritylation step. And so what this is referring to is removing this trityl group.
because right now this trityl group is going to prevent this from reacting further. And remember, we need to add the letters with this DMT on so that we don't get things like 2 G's added in a row. When we add these in, we're going to add them in that protected form so that we don't get that unwanted addition. And when we flood the reaction mixture with a bunch of these monomers, these individual phosphoramidites letters, that only one will get added on. Only one will couple at a time. And then we have the 3' end protected, remember, by this 2-cyanoethyl diisopropylamino group. And this is the part that's going to then, basically, this is going to be where you're going to attack. So you can think of it more like a sandal than an actual boot, because this part's actually going to get, attacked, and this is going to come off. So this part, this here, is actually going to get swapped out by the new letter. So we're going to activate it with like acidic conditions, make this a good leaving group. And now that the 5' OH of the group that is unprotected, we're going to deprotect this when we want this to react. And then this is going to react and attack this part, this phosphorus in the incoming letter, and kick this off. But then we're going to leave this part on until you're almost done, so that nothing else is going to get added there. And in the case of the first letter, this is actually going to be used to link this to a solid support so that you have things tied down. Remember, you don't have the help of polymerase holding things together. Instead, you're going to have this attached to like a bead or some sort of support. So that was the 3' position and the 5' position. And then we have the base. So we need to protect those bases, other than thymine, we need to protect these amine groups, which can be reactive. And so what we're going to want to do is we're going to actually protect them with various chemical groups. And here's a few examples of some that are commonly used. You often have something like benzoyl or isobutyryl groups, basically just preventing these from reacting when you don't want them reacted. And then at the end, we can deprotect them all at the same time. And remember then, with the RNA, you have an extra complication in the term of that 2' OH. And so you have to add extra chemicals to protect that group. Often this TBS, so this terbutyl dimethylsilyl. And so this you want to leave on until the very almost last minute so that you don't have anything react. And because that make this oxygen then go attack and cause chain breakage and all this stuff. And so you have these extra fancy groups that you have to have on the RNA. So most commonly that TBS, but this is kind of like a big bulky boot. So if you think about if you've ever had to be in one of those like walking cast boot things, it can slow you down. And so when you have a big bulky group like this, it can make the reaction go slower. So there are some alternatives that are more expensive, but can make things faster. This like TOM, this has a spacer, so it gets in the way less, as well as this ACE group, which is really super stable, so that you can actually order it still protected if you wanted to make modifications to it yourself, and then you remove it. And then there are ones that are removable under more gentle conditions, which you might need if you have various modifications to the bases. Okay, so now we've looked at the players, and now let's look at what's actually going to happen in this reaction. So remember, our general strategy is going to be protect the parts that we don't want to react and expose the parts that we do want to react and make the conditions so that they want to react. So starting with that first letter. So the first thing that we're going to need to do is, well, we're going to need to couple it to our support, and we're going to need to basically make it so that it's reactive. So remember, you have this DMT group. This DMT group is going to be protecting it so that it doesn't react at this 5' position, isn't free to react. But the first thing we need to do is going to make it so that it will react. And so it's going to react, remember, by kicking off this group. And so it's basically going to attack this phosphorus here, kick off this nitrogen-y group. And this is going to be helped by the acidic catalyst, ETT. And basically, this is going to make it so that this is a better leaving group, and this is going to be more favorable for attack. So once this attacks, this is basically what we call coupling, because you've coupled these two letters together. and they're coupled through this weird bond. So you can see it's kind of looks like one of those phosphodiester bonds that we've seen, but not really. And so we need to stabilize it. We're not going to, at this point, we're not going to turn it exactly into a phosphodiester bond because that would be too reactive still. We're going to make it into a more stable form of this weird thing. And so what we're going to do is we're going to oxidize it to protect it from like breaking and from reacting and all this stuff. But first we need to make sure that we stop any bad ones in their tracks before they react any further. So they use this capping step where basically you're going to put a lid on any of those monomers that were attached to the support, but that didn't get coupled. So they didn't have a letter added to them. If they didn't have a letter added to them, they're still going to have that free 5' OH. And if they still have that free 5' OH, well, now they can react in the next cycle, but they will have skipped a letter. And so that's how we got things like our weird, that's how we got things like our AC without that G. And so that's not what we want. So we need to cap them. And so we're going to cap them with a different group this time. This is typically an acetyl group for DNA or a terbutyl phenoxyacetic anhydride for RNA, something that's going to be really hard to come off. And so it's not going to come off in your various future cycles and therefore react. And so the capping is going to be that step. And now that we've capped that, now we want to go and we want to protect the right ones. We need to stabilize that bond between them, the bond that we formed in that coupling step. And so the way that we're going to do this is we're going to oxidize it. So basically we're going to take this and we're going to make it so that there's this double bond to oxygen here. This is going to basically be more stable. And now you can go and you can get ready to do the next cycle. And in order to do that, this is also going to be promoted by iodine in the presence of water impurity. And I'll also post a link to basically different blogs and papers and things like this with more information about the actual chemical details of these various steps. But just this is what we're looking at here. Is you have this oxidation step with iodine in this water pyrimidine mixture. So again, you just have all these, basically you can have these these molecules tethered down and then be flowing different chemicals over them to make things react in the way that you want. And so in this case, you're going to be oxidizing them, promoted by this iodine. So now what's going to happen is you have these two bases joined together and stabilized. And we want to now be able to do something with them. So we only have two letters together. We want to add more. And so if we want to add more, well, now we're going to need to take off this glove. We're going to need to remove that DMT. So we don't want to remove this cap from these, but we want to remove this original glove that we started with, this DMT protective group, so that we have a 5' OH that can then go and attack an incoming letter. So we need to remove that trityl group, that weird thing with that like 3 rings. That's called a trityl. And so we need to detritylate it. So we need to remove this part so that this 5' OH is free. And so this is going to be a detritylation step, just like we had in the beginning. So basically where we can use acid in order to get that to come off. Now we have that free 5' OH. And now what are we going to do? Well, what we could do is if we want to add more, we'll go through this cycle again. We'll couple in another amino acid. So we'll add another one of these phosphoramidite monomers, the letter we wanted added next. And then we'll go through this cycle over and over until the sequence is done. And so again, we don't have a template telling us when it's done. We just have some like computer program or something. So it's going to stop adding letters. It's adding those letters like one at a time, but lots of copies of that one. But then it's going to stop once they've gotten to the end of the sequence. And so the number of cycles you go through is going to be the number of letters in your sequence. And so once you get to the end, well, now you are done with the cycling, but you still have extra steps to finish things off, because there's a bunch of things that are weird about this molecule. It's got a bunch of protective groups, such as on these bases. It's got these weird bonds, so we don't have these phosphodiester bonds like normal. We have these weird things sticking off of them to protect them. So we need to deprotect all of these things in order to make our sequence like normal. And so, but in order to do that first, what we're also going to have to do is we're going to need to cut it off of the support that it's stuck on to. And so that support was great when we were making it, but it wouldn't be so great if you had to do like all your PCR reactions actually stuck to a plate. So instead, what we're going to do is we're going to cleave it off with this ester hydrolysis step. Basically remove this from the support. Now it's starting to look more normal, but what you're going to see is that you still need to deprotect things. So you typically use this like concentrated ammonia. So these really extreme conditions in order to take off those groups that we were using to protect things. Now we can make conditions where they won't be protected, because now we don't have to worry about things reacting. We just want to get all those protective groups off. One thing that you'll realize, though, is that you're actually going to have you're not going to have a 5' phosphate group, because remember, we never really had that 5' phosphate group. Instead, what you're going to have is a 5' OH. And so if you actually want there to be a 5' phosphate group, you're going to have to ask for it and pay for it. But that's the basics of this solid state oligosynthesis. So basically, you take your first base, you attach it to a bead. It has its 5' OH protected, so you need to deprotect that with this detrotylation step. You now add in your next letter through this coupling step. Those are going to get joined together, but they're going to get joined together in this kind of weird bond. And this is going to then need to be stabilized through an oxidation step. But before you do that, you're going to basically cap the ones that didn't react so that they don't react in the next cycle. Now you can go and you can continue on towards the next cycle by exposing the 5' OH on the letter that was just added, so that you can go through the cycle again and again until you decide you're done, cut it off from the support, remove those protective groups, and voila, you've got your almost normal oligonucleotides sequence. Now you're often going to want to clean up the products, because there are a lot of things that could go wrong, remember? And so at those steps, we were kind of making it so that we were removing the bad groups. We were doing that capping and things. So we're going to end up with a bunch of short mers. So these pieces that are the wrong length because you terminated them early, because they failed to couple in a cycle. And so there are various methods that can be used to purify the pieces you do want, such as HPLC, so high performance liquid chromatography. It's a column-based method, kind of like columns we use for protein purification, but there's different columns and there's different solvents and stuff. And then there's also page purification, where basically you can run like a page gel, just like we do in the lab, to separate nucleic acids pieces by their size. You can do this, but then actually cut those pieces out and purify them. Or instead of doing it in like one of these slab gels, they're often like capillary gels and things that are used for this purification purpose. And so I will post links to various resources that'll be helpful. There are also cool things that you could do when you're chemically synthesizing these, including using modified phosphoramidite. So if that wasn't modified enough for you, you can do things like have modifications to the bases and to various positions on them in order to get even cooler things that can then be used for various purposes. So to review this cell way versus the solid state way, and remember that the cell way is also going to be the way in your PCR tube, because that PCR tube is basically mimicking the way that your cells do it. In those cases, it's going to be template directed. So you have one strand being used as a template to promote the addition of the sequence of the other strands based on that template. In the solid state way, it's going to be directed by what you're giving them. So what's available. The letters are going to be held together by polymerase and base pairing. The sequence, the new letter and the old letter, are going to be basically held together by the polymerase and the base pairing. But in the solid state, you're going to have to bring them together. And so you're going to basically hold one in place. So hold the growing chain on the plate and then flood it with a high concentration of the incoming letter. These incoming letters are going to be added as phosphoramidite monomers rather than these nucleoside triphosphates that are normally added. Additionally, the way that it's added is that you have this free 5' OH that is going to make it so that you're adding in the 3' to 5' direction in the solid state way, as opposed to the normal 5' to 3' direction that happens in PCR and in your cells. In our cells and in that PCR tube, The groups get added correctly thanks to the polymerase, but we don't have anything to help make things happen correctly in terms of the actual addition step. So in your cells or in your PCR tube, the groups are going to be added correctly thanks to that polymerase. And in the solid state way, we have to use chemical tricks in order to protect the groups that we don't want to be reactive so that you only get addition in the places that you want. But then in order to make reactions happen in the places where you want, you have to deprotect those groups when you do want them to react. So there's a bunch of complications that have to go when you want to make things solid state, but this works well for doing short sequences, things like up to 60 or so nucleic, a couple hundred maybe nucleotides, or I guess not nucleotides at first. They're going to be phosphoramidite monomers, and then they kind of like turn into nucleotides. But these can be used for things like PCR primers and things like antisense oligonucleotides. So these short sequences, these what we call oligonucleotides, are typically made with this solid state synthesis using this phosphoramidite method. But remember how it's not that efficient and how each cycle you're going to be losing a lot of those duds. So you're going to have to be capping a lot of things. And each cycle, you're going to have to be flooding it with these different letters because you don't have the benefit of the polymerase and of the template kind of bringing things together. And so we need to, that kind of makes artificially high concentrations. And so we need to make actually high concentrations. And so we're going to be adding lots, and then we're going to be removing lots, and there's just going to be a lot of problems that could happen. And so you end up with a lot of that early termination. Bottom line, it's only good for short sequences. If you want a longer sequence, well, now you want to kind of have a template, ideally. And so you can use, if you have a DNA template, you can do things like you could use PCR if you just want DNA copies, or if you want RNA, you can do that too. You can use a T7 RNA polymerase is basically going to use a DNA template you give it in order to make the complementary RNA. And so there are ways that we can get this synthetic RNA to be made in a way that's not that solid state synthesis. So this is the way that's more like what happens in your body, but it's happening in a test tube. And so there are ways that we could do that for both DNA and for RNA. And this is going to be good for longer sequences. And it's a lot more simple because we're making enzymes do all the work for us.